Welcome to FDA Grand Rounds. We are very excited today to have Dr. Richard Beeger presenting on clinical omics biomarker discovery and validation in precision medicine applications. Dr. Beeger received his doctorate in biophysics from Purdue and completed postdoctoral training in nuclear magnetic resonance at Wesleyan University and Johns Hopkins University School of Medicine before joining NCTR's Division of Chemistry in 1998. He's currently the branch chief of the Omics Modeling, Imaging, and Chemistry branch in the Division of Systems Biology, a diverse group of scientists working in the fields of metabolomics, proteomics, tissue imaging, and chemical modeling of toxicological and disease endpoints. Dr. Beeger has been involved with metabolomics data quality since 2015 and was part of a group of scientists that started the Metabolomics Quality Assurance and Quality Control Consortium in 2018. He was on the coordinating committee of the Metabolomics Quality Assurance and Quality Control Consortium from 2019 to 2022, including serving as chair in 2022. Dr. Beeger won the 2016 Society of Toxicology Translational Impact Award for developing translational approaches to improve drug safety, disease detection and management, and human health. He has authored over 190 published manuscripts, including over 70 manuscripts in metabolomics, over 20 in chemical activity modeling, over 10 in proteomics and systems biology and personalized medicine, and he has 14 employee invention reports and 6 patents. So we are very honored to have Dr. Beeger with us today. And with that, I'm going to turn it over to Dr. Beeger. Thank you, Rich, for being with us.
Thank you, Rakesh, for the I'm very honored and humbled and actually excited for the invite and excited to present my research. As the title says, clinical omics biomarker discovery and validation in precision medicine applications. So anytime I say biomarker in this presentation, these are not biomarkers that have gone through full qualification at the FDA, although I do believe some of the ones I do present today could go through that. The disclaimer is presentation is my views and they do not reflect those of the US FDA. Any mention of commercial products is for clarification only and not intended for approval, endorsement, or recommendation. So my outline is to give a very brief introduction of the system biology, omics, and biomarker concepts, and then go through 4 examples, briefly go over 4 examples. And the first one will be a translational non-clinical metabolomics and microRNA biomarkers during acetaminophen toxicity. The second one will be discovery of proteomic biomarkers for the prediction of kidney recovery from dialysis-dependent AKI patients. And the third one will be the discovery and validation of baseline predictive cardiotoxic proteomics biomarkers observed in breast cancer patients receiving DOX treatment. And the last one will be a method development that used for discovery and validation of microRNA protein pairs associated with the severity of respiratory disease. This one is the last one is really going to be a method development, and it could be applied to many other applications besides respiratory disease. So systems biology is basically all the omics out there. We got epigenomics, genomics, transcriptomics, which has been used for me to look at the transcriptome, proteomics to look at the translation, and then metabolomics, and I'll say lipidomics to look at metabolites and lipids, and ultimately your phenotype. And all these omics environmental factors are going forward. And there's general omic level from left to right here, although we know in biology there's a lot of cross-spec and negative feedback. And over the years, many different technologies have been produced to look at each one of these omics. And ultimately what we're trying to do is find the phenotype, how to describe the phenotype, And it could be any combination of your DNA, RNA, proteins, metabolites, or epigenome. Uh, and ultimately, what the assistance biology is defined by NIH as an approved an approach for biomedical research for instead understanding the larger picture, be it the level of the organism, tissue, or cell, by putting the pieces together. And as I said, we're very good at taking pieces apart, but putting them back together is the bottleneck. And I'm going to describe some approaches for that today. So the Omics Biomarkers for precision medicine, as I said before, we have an exposome, your diet, your drug, your lifestyle, age, social interactions. Did you exercise? Are you stressed today for one reason or another? And even I would consider gut microbiome. Part of that is exposome. And this all goes into your human genome, your transcriptome, proteome, and metabolome. And ultimately what we want to define is, like I said, is a phenotype. And that phenotype could be your genotype. It could be your proteotype. It could be your metabotype. And all these phenotype capabilities are used to look at drug response phenotype. Does it have side effects? No side effects? Are you going to have a response or no response? And all this data can be put in to a situation where you're trying to really find biomarkers. And these biomarkers include toxicity and safety, PD, diagnostic, prognostic, predictive, and monitoring. So the Omics Biomarker flowchart, and here I like to step back. When I was given this invitation, I think they just said talk about omics. And so I've been, like you said, involved in omics for the last 15, 20 years. And so I really had to come up with an idea of what I could talk about in 45 minutes. And what I decided to do is only look at clinical studies and studies that where the Division of Systems Biology was involved with partially invalidation or hypothesis testing. And validation could be either analytical validation or looking at a different cohort. That being said, the Omics Biomarker flowchart is initially you have to have a lot of preparation for your samples. Which omics are you going to do? Are you going to do multi-omics? Are you going to do both open profiling and target analysis? This has to be decided early on. Different omics actually will need different types of sample tubes, and you have to understand this. And so that ideally, in the beginning, you want to aliquot all your samples so you don't have to freeze and thaw all your samples. Then you do omic analysis. Are you doing open profiling, or are you doing some target analysis, or some combination? And through this process, you have to be very involved with quality control. I'm not going to go into everything there, but we do the standard things that you need to do. We system suitability samples, pooled samples, mixing the samples together. When we've done the pooled samples, we have preset coefficient variation levels for allowing analytes or information to go forward into bioinformatics. In the end, bioinformatics and your data analysis really tailor to what biomarkers you're looking for. And you're going to be adding additional endpoints as clinical chemistry, histopathology, other endpoints from your patient group. And then ultimately, at the end, you're going to get identified pathways or multivariate biomarkers from the model. And this is just the discovery approach. And this is really, I really look at is something that you continually fine tune through hypothesis testing. So the first round you discover, second round you might screen a smaller set. And this is an iterative process until you get to the biomarkers you're really interested in use in the clinic. So I like to call it pharmacomics instead of pharmacogenomics, metabolomics, or proteomics. And just like pharmacogenetics, you can look at the baseline phenotype, and you can actually apply metabolomics or proteomics to build models to predict individual drug pharmacokinetic and other patient responses. And one of the examples, we will go over that. And then you can also look at during treatment and build models from the actual data or in between the PK responses and the ultimate response group of your of what you're studying. So I put this slide together in response to toxicity, and this is really something that was revised from what we published back in 2014 that was studying idiosyncratic liver toxicity. And in that study, what we did is we looked at liver toxicants and then control drugs that had no liver toxicity, and then a bunch of drugs that had idiosyncratic response. And we built models based on the control hepatotoxins in the control samples and looked at samples after the ADME process, but before the actual organ injury. So we were in that process of pre-injury, early events, and protection of cellular dysfunction without overt injury. And what we were seeing at that time was changes in lipids and bile acids. And since that's what the liver sort of is supposed to do, we were sort of seeing a very subtle changes in those before the actual organ injury. And for me, we're going to go through some of my examples, like the acetaminophen, example one, where we see an early event, and we'll discuss that. We're also in the acetaminophen, I'm going to talk about a biomarker that actually happens during the organ injury. In the second example, I'm going to talk about where you already have kidney injury, but now we're going through the repair and recovery process. In the third example, I'm going to talk about doxorubicin and potentially some pre-injury or early injury markers going on. And we're seeing these actually at TZR before they get dosed. We do have the data during that period in the pre-injury, but we're not ready to show it today. And then the final one, I'm going to talk about samples that are sorely in that pre-injury scenario and how to combine microRNA and proteomics. And what I believe the omics can really help heavily on is this pre-injury and the protection of cellular dysfunction without overt injury, and also in the recovery and adaptation process, because all these events are very multi-parameter events where the omics can give you that information. As it's well known, like some of the production events like glutathione conjugation, apoptosis, autophagy, all these things change over the course as you age, and they change in disease status, and not everybody changes at the same time. So understanding how these events protect you from organ injury, omics can help you on. So now I'm going to move forward and go through the examples. First example is the translation of non-clinical metabolomics and microRNA biomarkers during acetaminophen hepatotoxicity. This was a collaboration between NCTR and the Arkansas Children's Hospital Research Institute. So acetaminophen-induced toxicity causes oxidative stress, reactive oxidant species production, glutathione depletion, mitochondrial dysfunction, disruption of energy metabolism, liver necrosis, and altered immune responses. It's a frequent cause of acute liver failure in the U.S. and associated with hepatic necrosis. An early metabolomics NMR study by Cohen reported a disruption of energy metabolism during acetaminophen toxicity. And later, NIH utilized an LCMS-based metabolomics to profile serum samples from control acetaminophen-treated wild type and acetaminophen treated to the CYP2E1 null mice. And what they reported was accumulations of long-chain acetylcarnitines in serum of the APAP-treated wild type mice compared to control. And this is consistent with the disruption of fatty acid oxidation. So we decided to look at this. And why would you see beta oxidation increases in acetylcarnitines? And so here's the cartoon for beta oxidation. You have your fatty acid is combined with CoA through the fatty acid synthesis, FAS, and you get ACL-CoA. But this does not go into your mitochondria. It actually has to be converted to the acetylcarnitine before you can go into the mitochondria. And if the mitochondria has been damaged, the acetylcarnitines do not get into the mitochondria, and you see them into the biofluids like blood. So this will be the only loss and loadings plot I'm going to show you. But here we have a look at serum acetylcarnitines and a few other factors in B6 mice dosed with 200 milligrams per kilogram acetaminophen. And this is broken up by early time points for samples collected at pre-dose and then late time points. And so the three circles show you what's going on: late time points, control, and then late time points. And the loadings actually show that the long chain carnitines, palmitoyl, oleoyl, and myristoyl, cluster with the APAP adducts, where in the middle we just see carnitine and acetylcarnitine, and in the later endpoints we see glutathione and liver weight changes. And so the issue going from non-clinical to clinical is the response to toxins are time dependent, and that becomes the issue of when to find in the clinic. So to look at this, we collaborate with the the Children's Hospital and Laura James on an NIH-funded study that allowed daily urine and blood sampling of children and adolescents admitted with acetaminophen overdose. This was with 7 different other hospitals. It was an IB-approved study. And I'm going to discuss today 2 mechanistic biomarkers that we looked at, the acetylcarnitines and the microRNA 122. We did publish other papers on lipids and bioacids, and I left you the references there below. So the translational assistance response of palmitoyl carnitine and the acetaminophen hepatotoxicity is shown below. We have 3 different studies: the 200 milligram per kilogram acetaminophen in DICE, where we show the palmitoyl carnitine in blue boxes in ALT with the left-hand side showing the concentration in millimolar, micromolar, and then the ALT levels shown in red dots, and that's on the y-axis on the right-hand side. And as you can see, the palmitoyl carnitine increases around 4 hours and then drops to below control when the ALT is at the highest, they're around 8 hours. When we move forward to the rats, given 1,250 milligrams of acetaminophen, we see once again a very similar process where we see around the palmitoyl carnitine is increased early, around the 8 hours, and then comes back to control around 24 hours where the maximum for ALT occurs. And then in the human, many of the samples we got were actually treated by NAC before we got the samples. And so they were actually given the antidote and they were protected from the normal profile. One patient wasn't not diagnosed right away, and it showed a full profile of to the acetaminophen overdose. And there we see that the increase at 24 hours for palmitoyl carnitine, and then it drops off by 72 hours, and the ALT is increased at 48, 72, and 96 hours. And on top of that, looking at 3 different systems here, we actually analyzed palmitoyl carnitine in 3 different ways. We used a kit. I won't tell you what kit we used that anybody can buy in the mice study. And then on the rat study, we used the metabolomics LCM open profiling system, where we're now giving you just a ratio between the acetaminophen dose and the controls. And then in the human samples, we actually use a targeted LCMS system with a labeled standard in human. So palmitoyl carnitine represents a potential translational biomarker associated with mitochondrial beta oxidation dysfunction. Moving on to microRNA as biomarkers. So microRNA, they're typically about 22 nucleotides long. They're non-coding RNAs. Then altered tissue can indicate organ damage. MicroRNAs bind to RNA-induced silencing complex, known as RASC, to regulate translation. They can do it by two different ways, binding and stopping translation and regression, or they can actually cleave the messenger RNA. And one thing for biomarkers, they're actually much more stable in biofluids than messenger RNA. And some microRNAs are produced in a tissue-specific patterns. For example, microRNA 122 in the liver. So microRNA 122 in the clinical, it was first reported as a biomarker associated with viral and chemical-induced liver injury by Zhang and others in the clinical chemistry manuscript in 2010. And this started several groups, including our group, starting to look at microRNA 122. And then in the clinical study, we looked at how it compared to what healthy individuals that never received acetaminophen, ones that did receive a therapeutic dose of acetaminophen and the APAP overdose. And you can see there's an order of magnitude increase of the microRNA in the acetaminophen overdose group. When we looked at these across species, we did a 500 kilogram study in mice, the same 1,250 milligram study in rats in that same human APAP overdose patient. Here we're looking at the microRNA in 122 in blue, and the AL2 once again in red. And you can clearly see that the marker goes very similar to the ALT necrosis marker. So microRNA closely follows alanine aminotransferase ALT and appears to be a translational marker of hepatic necrosis in rodents and humans. So in conclusion, the long-chain ASL kinases were increased in rodents and children after acetaminophen overdose. It represents the beta oxidation capability and mitochondrial dysfunction. Doesn't necessarily indicate liver, but since we knew it was connected to the acetaminophen, that's a good prediction. There's temporal differences between the species and dosing, which makes going to the clinic different. The N-acetylcysteine treatment and the timing of the NAC treatment can affect the acetaminophen-induced liver injury biomarkers in the clinic. And that was one of the issues in this study. MicroRNA closely follows ALT and appears to be a translational indication of hepatic necrosis in rodents and humans. For the second study, I want to talk about the discovery of novel proteomic biomarkers for the prediction of kidney recovery from dialysis-dependent AKI patients. And this was a collaboration between NCTR, the University of Virginia, and the VA University of Pittsburgh and the University of Pittsburgh School of Medicine. So acute kidney injury requires dialysis. It's associated with prolonged hospitalization, mortality, progressive chronic kidney disease among survivors. Previous studies examined only select urine and serum kidney biomarkers for predicting the kidney recovery from AKI. In this particular study, we got samples from the VA NIH Acute Renal Failure Trial Network. They were going under renal replacement therapy, dialysis, and we obtained serum samples that were collected on day 8 of the therapy. And so they were under dialysis for 8 days or a week. And what we were trying to do is whether to predict whether they recovered before day 28 or they remained on the therapy for 28 days. And we used the SomaScan assay, and we looked at all the samples and found 119 proteins that were changed. The demographics for the study, as we can see, that the average age were in the 60s, and sex, congestive heart failure, diabetes, the sulfur score, serum calcium, and serum phosphate were not significant between the recovered and non-recovered groups. So I'll go over with the Soma affinity assay very briefly. Soma means slow off rate modified aptamer. And basically what it is just a chemically changed nucleotide situation where that mimics an antibody. And what this company does is they go through an 8-step process, and I'm not going to go through the processes here. And ultimately, what they do is they measure these on a DNA microRNA. When we started this study, they were on version, the technology came out around 2010. We looked at this, they were on version 3, which could allow you to look at 1,305 proteins in a sample. And this technology is rapidly improving. It has huge effects for precision medicine. Now, version 5 is available, and you can look at over 10,070 proteins. And what's more important, is now you can actually design your own 100 or 500 kits to look at different proteins. So you can discover using the 10,000 and now follow up with a version of 500 or 100 and screen those out going forward. And that's a huge improvement. That wasn't available when we were doing this initial study. So here, the volcano plot of day 8 proteins in patients that recovered kidney infection versus patients that did not recover by 28. Here we can see in green are the proteins, the 56 proteins that were decreased, and 53 proteins in red are shown that were increased. And as you can see on the decreased proteins, we can see inflammatory markers like FGF23, IL-6, IL-1R1, and CXCL16. And even a marker like NGAL, which is a kidney marker, went down. And we can see some chemokines increased on the other side. And what we did in this particular study is we classified the patients based on the 25 proteins most differentially expressed. And these markers went forward into further analysis using logistic regression analysis. But first we went into, and we took all 119 proteins and put them into a pathway analysis and looked at their biological functions and diseases. And in this particular view here, the bigger the square, the lower the p-value, the more significant it is. And the deeper the color, the higher the z-score of the function or the disease, where orange means is a positive z-score, and the blue means a lower z-score that has gone down. And what we see that the cell-cell signaling, cell growth, proliferation, cellular development, embryonic development, tissue morphology were all increased. And then the cell death and connective tissues apparently were decreased. And so that sort of shows that, this is representative of a patient improving. I'm not going to go through all the regression analysis. This is a partial list, so you guys can actually read it. The chemokine CXLC11 is shown at the top. They had the highest odds ratio of 7.29 and the univariate p-value of 0.0023. And at the bottom, I have the list of the few ones that actually had smaller odds ratios less than 1. So we have the CXCL16, IL-6, and FGF23 that were lower and significant. We did both univariate and multivariate, and the odds ratios in the p-values were actually quite similar. And so what we ended up was a set of biomarkers that were associated with better kidney, and there's chemokines that were increased. And now the ones that decreased, we have four things that are going down there involved with inflammation: CXCL16, IL-1R, IL-6, and FGF23. And static calcin 1 is actually an acute injury biomarker for kidney. So we are very comfortable with this. Very pleased to see this result. It actually was quite consistent with a previous study that we did where we saw FG23 and IL-6 were specifically associated with mortality in the AKI dialysis patients. And actually, FGF23 has actually been reported quite often as a poor prognosis marker. In fact, that we're seeing it go down is actually also very comforting. And we actually found it by discovery is also interesting. So the correlation between day 8 soma scan and Olink. So how do we validate these markers? And so this was a problem back then. It was you have this huge array of proteins, and how do you actually go through it? At that time, all we could really do is go into Olink, and they had an inflammation panel that had 93 proteins that were measured by Olink using their toxicity extension assay. And here we looked at the correlation between 3 proteins and got a correlation coefficient, a p-value. And the 3 that we were able to get was the CXC motif chemokine 11, which was the highest increased protein. And then we also saw the interleukin 6 and the fibroblast growth factor 23 that were the odds ratios that went down. And we saw significant correlations for all 3 of them. Ironically, just very recently, Olink now just released what they call the Olink Focus assay, where you can actually request and have a design kit to look at 21 proteins right now, which would have been perfect for this study. It has a pretty large upfront cost, but it would have been able to assay all 21 instead of just 3. So the SomaScan results suggest protein changes between survival-related proteins and chemokines in regulatory angiogenesis, endothelial and epithelial remodeling, and kidney cell regeneration involved with the kidney recovery during dialysis. Protein decreases in inflammatory proteins were more significant increases than kidney injury markers. So I showed you NGAL, but it didn't end up into our regression analysis. We only were able to validate 3 proteins using the correlation between SomaScan and Olink. Obviously, these markers have to go through another iteration, and additional studies are needed. Going forward into our example number 3, I'd like to talk about the discovery and validation of predictive proteomics cardiotoxicity biomarkers that are observed in breast cancer patients receiving DOX treatment. And this is a collaboration between NCTR, University of Arkansas Medical School, CEDAR, and CBER. So toxin-induced cardiotoxicity, it can manifest as an early onset acute toxicity, subacute toxicity, or a late onset toxicity. And the late one is where the issue is, and it really depends on the cumulative dose. And so generally, there's an incidence of less than 3% on patients receiving a cumulative dose less than 250 milligrams per meter squared. But the risk goes up to 20% when the dose exceeds 500 milligrams per meter squared. And the current challenge is the preclinical models are not that predictive going into the clinic. And this may be just due to the docs and the markers and the timing issue. The cardiotoxicity and the studies have shown that oxidative stress, myocardial dysfunction, inflammation pathways, and cell death can occur going forward before heart failure, and that could be with the conductivity failure, myocardial fibrosis, and metabolic reprogramming. And what I'm going to talk to you about is about early detection. And in this particular case that I'll be able to show you is actually before the dosing. And so it could actually be used in risk stratification. Current biomarkers of cardiotoxicity, and what they've done is an overview here. The left ventricular ejection fraction, LVEF, cannot predict late cardiotoxicity. Ultrasensitive troponin has a predictive value of about 44 sensitive. Large trails are still needed. Cardiotroponin, it predicted diastolic dysfunction in 34% of patients. It's controversial, not well established. NT-proBNP is controversial, not well established. MPO has a probability of predicting is only around the 24 to 36 range. There's a number of proteins like CRP, GDF, PIGF, SPFLT, and galactin, GLL have no predictive value. One that has a little bit is peak systolic myocardial strain that has a positive predictive value of 53%, and the sensitivity and specificity of 74 and 73, respectively. Large trials are needed, and you have to actually go through and decide whether you're doing longitudinal or segmental. So our study design is we had a inclusion criteria of 18 to 100 years old, and they're not pregnant or breastfeeding. They have cancer, they required retreatment with docs, and they were willing to participate. Exclusion was they were outside that age group, they were pregnant or breastfeeding, they had prior history of radiotherapy or chemotherapy with docs, and they declined. And so the study design is shown in the lower left-hand corner, where they received 60 milligrams per meter squared of docs plus 600 milligrams per meter squared of cyclophosphamide. And they were given at 4 different time points. We did a multi-gated acquisition scan to measure the left ventricular ejection fraction at T0 before the first dose and after 2 or 3 weeks after the 4th dose. Blood draws were taken at T0 right before the first dose, T1 right before the second dose, and T2 right before the second dose. Our approach on these T0 samples, the baseline samples, is what we did. We had a biomarker discovery group where we used the SomaScan on 1,305 proteins. And then we had a biomarker validation group where we did the Olink assays, and we looked at 1,463 proteins. Our criteria for cardiotoxicity in model development was the left ventricular ejection fraction had more than a 10% absolute reduction. Now, many of these patients had that. I'd like to inform everybody here that everybody did recover. Our discovery set had 39 patients. Our validation set had 44 patients, and we used PLSDA modeling. And then we evaluated its performance in these both in the discovery and validation sets. So the demographics for the discovery group, we typically were in the 50s. BMI was basically overweight, and baseline was one area there where we saw an increase in the baseline left ventricular ejection fraction, which was unexpected and was predictive. And there are reasons why this might happen. We put this into our revision of the manuscript. And that was observed in both the discovery and the validation cohort. But we didn't see any changes, whether it was ER positive or triple negative or hypertension in either group. So at first here I'm showing you the volcano plots and the discovery of the SomaScan and the validation only. And here I've highlighted some of the proteins that increased in red and decreased in green that were overlapped between the two. And here we actually decided only to look at the overlapping proteins. There's 5 actually, even though we're measuring over 1,000 in both cases, there was only 579 proteins that overlapped between these two technologies. And so there was a few biomarkers that we discovered and we tested in the validation set. And to validate these, we actually, when we sent out for Olink we did send another 25 samples from the discovery set and looked at the correlation that were not used and informed. The only thing we used it for is to look at the correlation of those markers in the SomaScan and only get T0. And so that the 6 markers that we decided to go for was bioglycan, chitin A, 5, CA6, CD109, cystatin F, and TSP4. And all these show a significant correlation. The only one that was a little fuzzy is the bioglycan with a correlation of 0.45, but it was still significant with a p-value of 0.01. So why would these markers be useful and their potential roles? So bioglycan, also known as BGN, in the discovery set was increased by a factor of 1.34, in the validation set of 2.77, and the p-values were 0.03 and 0.02 respectively. And this is a component of the extracellular matrix and may contribute to the cardiac ECM remodeling. In fact, 5 of these proteins are all involved in ECM remodeling. The only one that's not is the CA hydroxylase CA6, and that's involved in pH regulation, where it's known potentially if you get a very acidic situation in the heart, that could be problematic. And so we see similar results going from the discovery validation set for Caterin 5, CDH5, with an increase of 1.29 and 1.28 in the groups. And this could be overexpressed during ischemic and dilated cardiomyopathy and associated with the progression of chronic heart failure. CD109 was increased by 1.77 in the discovery set and 1.23 in the validation set. And this could be associated with the valve disease. The thrombosin 4, TSP4, increased by 1.44 and 1.5, and this is a component of ECM. It may respond to cardiac pressure load and contribute to hypertrophy and heart failure. In fact, hypertrophy is actually one of the potential reasons we were actually seeing the increase in the left ventricle ejection fraction. And the only one that did go down was the cystatin 5, and that is involved in ECM remodeling, and it's involved with inhibiting existing cataspins. So an overview, in our discovery set, we had 39 patients that we went into a SomaScan assay. We found 5 proteins that were increased and 1 that was down, plus its baseline level of left ejection fraction. We went into a therapy. We went into a machine learning algorithm, PLSDA, and did classification of non-cardiotoxic versus treatment-related cardiotoxicity. We took that model, PLSDA model, and evaluated it in a validation set of samples with 44 patients. And we looked at those same 6 markers. 5 of them went up again and 1 went down, plus the baseline left ventricle ejection fraction. Used the same model, the PLS-based predictor model. In the end, we had a sensitivity of 80%, the specificity of 82%, and an accuracy of 86%. So we've actually now gone forward and received approval from OAWH to start another study called the Informed Prediction of Cycling-Associated Cardiotoxicity, which we call IMPACT. And this is a multi-center biomarker qualification study. We were getting samples from the Beth Israel Medical Center. We received all those samples, the School of Medicine at the Washington University. We've received the majority of those samples. We're still waiting for a few more. And we're still waiting on the University of Arkansas medical science samples. And once we receive all these markers, these will be sent out for analysis and tested whether these 6 markers work in there. One thing is we're also expanding more than just breast cancer in these samples. So it'll be interesting to see if it only works in breast cancer or obese breast cancer. And these are samples that came, once again, before treatment. So finally, I'd like to discuss about the method development used for discovery and validation of microRNA protein pairs associated with the severity of respiratory disease. This is a collaboration between NCTR, the University of Tennessee Health Science Center, CEDAR, and the University of Arkansas for Medical Sciences. So respiratory disease impact on human health, it causes high mortality and morbidity. It's one of the leading causes of death. The physical functioning, chronic illness causes severe persistent symptoms of cough, chronic mucus production, breathlessness that limits physical activity. Long-term complications of advanced lung disease often leads to complications like pulmonary hypertension, heart failure, respiratory failure. Risk factors could be older and younger people, the other two, and those with underlying health conditions that are higher risk for severe diseases. And the goal of our study was to collect and analyze microRNA profiling and proteomics to discover potential system biology, mechanistic predictive markers for the development of severe respiratory disease. And so here is the SOP that we developed. We sent out, in both cases, the samples for measuring microRNAs to a group called Pyrolysis, and they used the HC platform, and they were able to send us back data on 2,083 microRNAs. We sent out the other samples to Omics, and we were now at a higher version 4, and there we're measuring 2921 proteins. We had already sent, and when we sent these samples out, we sent pooled samples. They used this for our QCs and coefficient of variation limitations to come out with a differentially expressed microRNAs, DEMS. And then we did the same thing in pool C's to end up with a differentially expressed protein for each response group. We did this in the discovery cohort. They had a mild group, a moderate group, a severe group, and ages slightly increased. The gender was pretty even, and in the race Black, it was higher prevalent in the discovery cohort with the severe group. In the validation cohort, it's sort of reversed. The Black was more in the mild group. We do see a slightly different age, a little bit older in the validation cohort. But in the validation cohort, we only looked at the biomarkers, the DEMS and DEPs. We did not do we didn't look at for new DEMS or DEPs there. And so once we have the DEPs, what we do is we put these into pathway analysis, just using the DEPs. And then the key here is how to connect the DEMS to the DEPs. And so we did this 2 different ways. In one way, we looked at the microRNA messenger RNA, which we use as a surrogate for the protein associated. We did use prediction software to do this in databases to look at this. The other way was just to go at a direct correlation between the microRNAs, their DEMS, and the protein pairs. And I'm just going to show you with the correlations that have greater than 0.05. And when I put the absolute sign there, most of these are negative. And so we're only going to be talking about the negative correlations going forward here. And so in the end, it's to put all these things together, your DEMS to your DEPs to your pathways. And I'm going to show you 2 different Sankays, one where we actually have an idea of the connections between the messenger RNA and the protein and the pathways, and then the correlation Sankey, where these are just correlations. We don't know exactly why they're connected. So to go through how the process of looking at the microRNA messenger RNA binding mechanisms, it can happen by an imperfect complementary. That is, the messenger RNA can bind to the 3' end of the target messenger RNA, and with a partial matching, and this will cause a translational repression. In some cases, you get perfect complementary, and this could lead to a messenger RNA degradation. Most of the time, this occurs on the 3' end. There are functional sites that also exist on the coding regions on the 5' end. But the thing here is the outcome of both situations result in downregulation of protein translation, either by preventing the protein synthesis or reducing the messenger RNA stability. So if the scenario is if you have low microRNA, the protein it normally regulates will be overexpressed, and you'll see a high levels there. If the microRNA is high, its protein will be underexpressed. And so microRNAs are acting like a drug. In fact, actually, right now, there are many papers out there talking about microRNAs as drugs or microRNA mimics as drugs. So for the prediction of microRNA binding to microRNA, what we used was IPA, and it uses a target scan content that we're using predicted what are the relations from that. And then the predictions are either high or moderate conference. It uses targets from mammalian microRNAs. The targets are predicted on an algorithm by searching for the presence of conserved 8-mers and 7-mer sites that match the seed region of each microRNA. We also looked at content in the tarbase that have been experimentally demonstrated for their connections, and MRI records, which also has experimentally validated human rat and mouse microRNA messenger RNA interactions from hundreds of published articles. And the IPA database is curated by experts, and I can't say how important that really is going forward. Yeah. So what I'm going to show you here is really the Sankey. We actually had to limit this to higher levels of fold change and higher level of p-values to go forward. But here it shows the Sankey of the microRNAs connected to their proteins and the pathways going forward. And there's actually, in this particular one, the green lines coming from the microRNAs to the proteins have been experimentally observed in other studies. And the orange lines here are highly predicted, and there was 94 microRNA connections here. And here we're only showing severe versus mild combinations. In every connection is a biomarker, and every connection is a negative regulation. So that either the protein is increased and the microRNA is down, or vice versa, and there's only a very few cases of that. And this diagram shows 60 microRNAs, 58 proteins, and 29 pathways. So how do you test this? And well, we use the validation cohort to actually look at the same set of pairs in the training and test set. And so in this particular case, the average correlation of these predicted pairs was in the discovery set minus 0.334, and actually in the validation cohort set, very similar of minus 0.339. And here we're showing a map of all 122 microRNA protein pairs. And the line shows that we're showing 122. There's a correlation between all these of 0.43 with a p-value of less than 0.001. And the ones that are far over to the lower left-hand corner are the ones that I would take forward in a future study. And below this, I'm showing you one right now is microRNA 146A5P that was negatively correlated to S100 calcium binding protein, also known as S100A12. In the discovery set, it has a correlation of of minus 0.47 and a p-value of 1.86 10 to the minus 6. And in the validation cohort, we have a minus 0.63 and a p-value of 10 to the minus 11. And this might be related to the neutrophil axillary traps that are produced that may contribute to thrombic complications. One thing that I was very interested in this, so around 0.2 is where the correlation has a p-value of 0.05. So we are seeing some that we predicted aren't actually aren't significant. And I also see up here in the upper right-hand corner ones that both data sets saying probably we're not seeing any relationship between them. They're very close to zero. And these might actually be false positive prediction, positives from the prediction algorithms. One of the things that we actually did, as I said, we looked at the correlation Sankey. And here we see a much larger, robust correlation. Here, before I showed you 122, here we're seeing 912. And these are just from p-value less than 0.05 in the severe versus mild with a protein correlation greater than 0.05 in the training set cohort. And previously, I showed you 112, and only those 5 green lines and 12 orange lines, which you can't see, were in the previous slide, or 2 slides before, in the Sankey before. And now we have in blue, which dominates the connections between microRNA and the proteins, that represents proteins that have no predictions on the microRNA protein association are available. But in the validation cohort, we still see a greater than minus 0.4 correlation. There was, and this represents about 2/3 of all these things going on. And then the gray lines represent the other third, where we have no association, and the validation was, the correlation was less than 0.4. There are 64 microRNAs and here 55 proteins and 25 pathways. There are a lot of very interesting correlations here, but since we don't know what's causing them, I'm not going to go over that. There's probably, a lot of chance correlations in here, but we're probably missing quite a few false negative predictions from the IPA prediction of IPA microRNA, the messenger RNA. So the microRNA protein analysis conclusions, we developed a method that can integrate microRNA profiles and proteomics data. This method was discovered in this particular case, 519 DEMs and 700 DEPs that were significantly associated with respiratory disease severity. The DEMs were predicted to messenger RNA protein surrogates to DEPs to form marker RNA protein pairs. A significant correlation was found between these 122 pairs in the training cohort and the same 122 pairs in the test cohort. These findings provide mechanistic insights to messenger RNA regulation of host responses to respiratory disease that could use in precision medicine decisions and identify potential care. But I truly believe that this could be used in many other diseases, cancers, and other type potential medicine decisions. So the predictive omics biomarker conclusions, you need to undergo analytical validation. This is an issue in the proteomics world. Excuse me. Hay fever is starting to catch up to me. We do have yellow fever going on here in Arkansas. And we try to overcome that by looking at different kits. You have to go through biological evaluation in multiple cohorts before you can get to something. We were able to look at the markers in baseline and treatment samples. The issue is there's different temporal responses in non-clinical studies and clinical studies. That becomes, how do you go forward with that? And microRNA and proteomic analysis can provide information on the messenger RNA regulation of protein expression. Some of the challenges there is quality control and reporting. And as I said earlier, I've been involved with MQAC since it started over 10 years ago, MQAC. There are also the quality control for proteome, the human proteome, and the lipid standards. I didn't talk about lipids a lot here, but they're going to be quite useful going forward. The one thing is all these are highly MS-based. And now what we're seeing now is in the proteomics world, these array-based things, that the Human Proteome Organization Standards Initiative doesn't really look at the array-based standards. And so really now the only people really looking at these things are the vendors themselves. And I do think this is indeed something that's going to be a wider look at by more than just the vendors themselves. The biggest issue is managing and analyze vast data volumes. So the combination of the proteome and the microRNA in two sample sets was huge in keeping it going forward and looking at this heterogeneous data and being able to analytically and biologically validate this data. We used IPA curated databases. There are other databases that aren't quite as curated, and you could end up with slightly different answers if you use other databases. I think this is an issue in the omics world, is using validated data, and you have to go into validated databases to do that. Model validation and AI are obviously, AI is going to play a huge role in multi-omics and precision medicine. And once again, I'm going to have to say AI better use very validated data and databases. Then training professionals to interpret the omics data and the AI models, that's going to take time. Ensuring data privacy. Proteomics data is not as bad as DNA, but you can, if you have a large enough data set, start figuring out who people are. And then there's high costs. The funding for this was the FDA Office of Women's Health that provided funding for all three doc studies I talked about. The CEDAR funding came for the respiratory disease. NCTR provided funding for all the things I talked about. The VA had provided funding for the AKI. UAMS was involved with the API, the docs in respiratory disease, and UTHSC was involved with the respiratory disease. Thank you for your attendance.